Welcome. This is the module of AST3114, Integrated Science Project. In this video, we are going to talk about the Project 2 with the topic of protease in natural fruits, such as kiwi fruit and papaya. Before the start of the project, let's talk about the steps of the scientific investigation. The first step is observation. You need to observe the things around you in daily life. And the second step is identify problem. In this step, what do you want to find out? You often start to raise a question. And for the third step, propose hypothesis. You should make a smart guess of your project. After that, step 4, make predictions. In this step, you should predict what will happen and why this will happen. And step 5, design experiment. In this step, you need to design a suitable experiment to test whether the hypothesis is correct or not. And you need to identify the variables, such as independent variable and dependent variable. And then determine the apparatus and what materials is required. Moreover, you need to determine the procedure of the experiment to collect and analyze data. Step 6. Perform experiment. In the step of performing experiment, you need to perform a fair test in which consists of 3 C's. Change, constant, and compare. You only change the variables under investigation. All other variables must be kept constant and compare the result. In addition, you need to do the risk assessment. And finally, observe carefully, record results accurately. Step 7. Collect data and record results. You need to write down what has been observed, such as measurements, color change, behaviors, and descriptions. And the result will be presented as word, diagram, photos, video, table, or graph. For step 8, draw and evaluate the conclusion. And the final step is reporting. You need to write the report for what you have investigated. And in this case, we observed kiwi fruit and papaya can be used as natural meat tenderizer. Then we will raise the questions that why kiwi fruit and papaya can tenderize meat? And is there anything inside kiwi fruit and papaya to tenderize meat? And the answer is yes, there is presence of protease. Protease can break down proteins into amino acids. After that, we can set up the hypothesis according to the observations and questions. And the hypotheses are kiwi fruit has better tenderizing ability than papaya, and kiwi fruit contains high amounts of protease to digest protein in meat. And finally, we can perform a fair test with 3 C's. First, we change the type of fruit. And second, we keep the amount of food as constant. And then we compare the tantalizing ability. Let's go through the information search process. There are 6 steps. Step 1. Initiation. We recognize the need for new information. Step 2. Selection. We decide what topic will be investigated and how to proceed. Step 3. Exploration. The information on the topic is gathered. Step 4. Formulation. We start to evaluate the information that has been gathered. Step 5. Collection. We know what is needed to support the focus. And the final step, search closure. We summarize and report the information. Let's talk about the introduction of this project. Enzymes are biological catalysts that speed up chemical reactions. The substrates are bound to the active sites of the enzyme, and the enzymes turns the substrates into different products. And we can see that enzymes is not used up and can be reused. And in a digestion process, proteins are broken down into amino acids by the actions of enzyme called protease. In this project, scientists can use a milk agar study to demonstrate the activities of protease. Actually, milk agar is a mixture of milk protein with nutrients agar in a solidified form. And the milk agar is white in color because of the milk protein. In the presence of protease, proteins will be digested, turning the white color into transparent. This transparent area appeared is called a clear zone. And we can obtain the area of clear zone by using this formula. The objective of this project. And we can set up the hypothesis as among kiwi fruit and papaya, the amount of protease in kiwi fruit is higher than papaya. And the independent variable of this hypothesis is the types of fruit. And the dependent variable of this hypothesis is the area of the clear zones or the amount of protease. The resources of this project. And we can see the pictures of the materials and apparatus. And here is the list of the resources. Let's go to the procedure. And this is the procedure of preparation of the milk agar plate. And we'll show you in the video. This is the agar powder. Which weighs about 2.8 grams of agar powder.
And this is the milk powder. Weigh about 2 grams of milk powder. Dissolve the milk powder in 20 ml of distilled water. Add the egg powder in 80 ml of boiled distilled water. Heat the mixture and stir frequently to dissolve the egg powder completely. Transfer the milk solution to the hot egg solution. Stir to mix. Pour the resultant solution into several petri dishes. Replace the lips and allow the place to cool and set.
And here is the procedure of preparation of the fruit juice. Let's watch the video again.
Cut the kiwi fruit into small pieces. Roughly grind the fruit pieces by mortar and pestle. Filter the kiwi fruit juice by glass. And collect the kiwi fruit juice into a beaker. Cut the papaya into small pieces. Roughly grind the papaya pieces by mortar and pestle. Filter the papaya juice by glass. Collect the papaya juice into a beaker.
And this is the procedure of determination of the clear zones. Let's watch the video again.
This is the milk agar plate. Gently press the copper down into the milk agar to make 3 wells. Replace the lid. Label the wells A, B, and C by marking on the back of the petri dish. Use the dropper to fill up the wells A, B, C with kiwi fruit juice, papaya juice, and distilled water, respectively. Replace the lid and incubate at room temperature for 1 hour.
Now we can use the camera to take the pictures of the result. Let's see the result. Here is the Protease. The result showed that kiwi fruit juice gave larger area of clear zones than that of papaya juice, and the distilled water gave no observation change for the result. The results are then showed as a bar chart. It shows that kiwi fruit juice resulted in a larger areas of clear zones than papaya juice. Since the area of clear zone is directly proportional to the amount of proteinase, the results reflect that kiwi fruit contain higher amounts of proteinase than that of papaya. And the hypothesis was supported by the experimental result. Let's move on to discuss the experimental result. For the rationalizations of the results, proteinase in the fruit juices diffuse to the milk agar and catalyze the breakdown of the right milk protein. And therefore, clear zones are formed around the wells containing fruit juices with proteinase. And it is assumed that the chemicals in the fruit juices would not affect the formations of the clear zones, and the diameter of the clear zones has a positive correlation with the enzyme activity. And it should be noticed that water is used as control in order to confirm that the formation of clear zones is due to the fruit juices. After that, let's analyze the errors in this project. The method of measuring the diameter of the clear zones was probably not very accurate because the shape of clear zone cannot be a perfect circle, and therefore the calculated area of clear zone might not be proportional to the amount of protease in food. And so, this may cause errors in comparing the amounts of protease in different foods. And for more errors, you can think by yourself. For the suggestions for the improvement, we can place the milk agar plate on a piece of graph paper and examine against light to measure the diameter or area of the clear zones around the wells. And for more improvements, you can also think by yourself according to what errors you have analyzed. For the risk assessment, before talking the risk assessment of this project, let's talk about the 5 steps to risk assessment. Step 1. Look for the hazards. And step 2. Decide who might be harmed and how. Step 3. Evaluate the risk arise from the hazardous materials or procedures. Step 4. Record your findings. And the final step is review your assessment. For the risk assessment of this project, first, the glass is fragile and may break, we should handle with care. And second, we may get heat burn from hot water, we should handle with proper protective gloves. And third, we may get injured from improper use of life, we should handle with care too. You can think more risk by yourself. Let's move on to the conclusion. And for the conclusion, it should include first, analysis and interpretation of the data. And second, finding a meaning for what has been observed. And third, deciding whether the hypothesis is supported or rejected. And the conclusion of this project is that the hypothesis among kiwi fruit and papaya, the amount of protease of kiwi fruit is higher than papaya, was supported by the experimental result. And also, the kiwi fruit juice resulted in a greater area of clear zone from the milk agar plate than that of papaya fruit juice. And finally, the references. Here is the references of this project. Thanks for watching. If you are encountering any problems, please feel free to contact your module lecturer